Now let's switch gear from inspiration to information. For many of us, the diagnosis of ZTTK came with the flood of medical terms, acronyms, and technical reports that left us feeling overwhelmed. If you've ever read a genetic test result and felt more confused than empowered, this session is for you. We're lucky to be joined by Raymond Belanger Deloge, a board-certified genetic counselor and rare disease research specialist at Boston Children's Hospital. Ray works at the intersection of families, science, and communication. His job is literally to translate genetics into human understanding. Today, he'll walk us through some of the core genetic concepts that help us understand ZTTK, what the gene does, what happens when it's altered, why it matters for our children, and the treatments we're trying to develop. Please welcome Ray Belanger Deloge on stage.
All right, how's everybody doing? First thing in the morning, hope everybody's got their caffeine, feeling awake. Thank you, huge thank you to Ada and Nathan for inviting me here to speak today. It's a true honor, and congratulations to you all. Having your kickoff conference is a huge accomplishment, so really enjoy it, get the most you can out of it. It'll be a great experience for you all. My name is Ray Belanger. As Ada said, I'm a genetic counselor, specifically on the research side, and I work in the Rosamond Stone Zander, or RSZ TNC, at the Boston Children's Hospital. There's going to be a lot of acronyms. I'll try to define them all. Right out of the gate, we get an acronym, but all right. So my job today is to kind of go over some basic foundational information, both about genetics and also kind of the research process for neurodevelopmental disorders, or NDDs. I want to start off with just a couple of questions. These are more geared towards the families in the audience. How many of you had any biology or genetics in school that you can remember? Just a quick show of hands if you'll entertain me. Okay, great. Ada hinted at this. Genetics can be kind of an overwhelming topic, so put your hands up again if genetics is something that you feel is kind of overwhelming or really confusing. Not to put everybody on the spot. Okay, great. So that's why I'm here today. I hope my talk can kind of simplify things a little bit, make genetics something that's a little bit more palatable. So yeah, let's dive in. So I want to start with just the basic biological pathway of genes, genomes to protein. So inside of our body, we have trillions and trillions and trillions of cells. And inside all of our cells is our DNA, or our genome. A chromosome, you may have seen pictures of chromosomes on something called a karyotype. These are compacted DNA that contain strands of DNA. And within those chromosomes are our genes. And a gene is a segment of DNA that encodes a protein. And proteins, we know we have many of them. They perform a wide variety of tasks in our body. And if we look at ZTTK and SON specifically, we know that the SON gene is located on chromosome 21. And the SON gene encodes for the SON protein. On a very basic level, we believe that the SON protein is important for regulating other genes. We inherit half of our DNA from one parent and half from the other parent. We get 23 chromosomes from one parent, 23 chromosomes from another parent, come together to make 46 chromosomes that provide the blueprint for our bodies, how we grow, develop, things like that. And one way to simplify things, an analogy that I like to use is that we can think of our genome like a cookbook. Our genome refers to the entire cookbook, from start to finish. Chromosomes you can think of as chapters in the cookbook, certain sections. So you get your pastas, you get your salads, you get your soups. And a gene is like a single recipe in the cookbook, and the protein is the output of that recipe, or what you end up cooking. And I mentioned that we inherit our genes, from both of our parents. So that means, things can be passed down from generation to generation. And we see this happening in a couple of different ways. We call these inheritance patterns. And the one that I want to focus on today, because it's relevant for ZTTK and how it's passed down, is called autosomal dominant. And on the screen behind me is a tool called a pedigree. You'll see a lot of genetic counselors, geneticists, other doctors use these in clinic. You may have seen one drawn before. But these are tools to help us track specific traits or conditions in a family. So you'll see that. Let me grab the pointer here. Okay. Squares are males, circles are females, and we have the different lines of the generation of the family. So this is a 3-generation pedigree. If a circle or square is filled in, like these here, we know that this person is affected with either the condition or trait that we're interested in. This group of individuals here are siblings, if you have the horizontal line here, and then shapes with the lines that kind of go through the middle of them, these are mating pairs. Now, autosomal dominant inheritance is kind of characterized by, traditionally you'll see multiple individuals in each generation affected with, the trait or condition that you're interested in. But pedigrees more often look like this, especially if we're considering rare diseases. And that's because many rare diseases are caused by what we call de novo genetic changes. You may have heard this word before, maybe in your genetic counseling appointment if you had one. But de novo basically refers to a genetic change that was not inherited from either parent. It was something that happened, randomly in the affected individual. And it's not uncommon, especially in the clinical sense, that we hear from parents that they learn of these de novo changes. Their biggest question is, why did this happen? Why did this happen? Was it something that we did? And I want to say that it's not uncommon to feel guilt or shame or think that it was maybe your fault. This is completely not true. De novo changes happen randomly. There's nothing that either parent did or didn't do during preconception, during the pregnancy that would have led to a de novo change. And that's because, our genes or our DNA has many, many changes from generation to generation. And not all of these changes are leading to a genetic condition, which I'll talk more about in a little bit. Now, looking at reproductive risks, because ZTTK is an autosomal dominant condition, we know that each child of a parent with ZTTK has a 50% chance of inheriting ZTTK. I know it looks pretty simple on the screen here. It doesn't mean if somebody who has ZTTK has 4 children, that 2 of them will have the condition and 2 will not. That 50% chance resets with every pregnancy. So it's possible that somebody could have multiple children and all of them are affected or none of them are affected. Again, that 50% chance resets with each pregnancy. And I keep talking about genetic changes, and what I mean by this are genetic variants. That's a term that we like to use. You may have heard this before. And there are multiple different types of genetic variants, and there's 2 that I'll focus on today. The first being a truncating variant. If we stick with our recipe cookbook analogy, we expect a sentence in a recipe to read, add 1 cup of flour. A truncating variant is one that stops the sentence in the recipe too soon. You'll see the example on the screen. And typically when that happens, you don't end up with what you're cooking. You don't end up with your protein. A missense variant is a little bit more nuanced, and that is just a 1-letter change in that sentence. So instead of the sentence saying add 1 cup of flour, we have add 1 cut of flour. And these single letter changes can lead to, for example, on the screen, we have cookies. Your cookie might not look exactly right. It may not taste exactly right, but you may still end up with a cookie or a protein with the missense change. And how do we detect genetic variants? We have multiple types of tests that have strengths for different types of genetic variants. Genome sequencing looks for changes in the entire cookbook. I don't know if you've read cookbooks, look at recipes online. Oftentimes you have to scroll through somebody's entire life story before you get to the recipe. It's very annoying sometimes if you're trying to make something quick. But genome sequencing will look at all of that content from start to finish. Exome sequencing just focuses on the actual recipes, not all of the other content in the book, not all of the other DNA. Gene panels are types of tests that look at genes that have similarities or they cause similar conditions. So for example, you'll see commonly gene panels for things like hereditary cancer, different hereditary heart conditions, things like that. And microarrays focus on larger types of genetic variants. So these could be large deletions, large duplications. So if you think of the recipe book, think of maybe a page or two that's been deleted or been duplicated. But in the rare disease world, the ones you've probably heard most about are genome sequencing and exome sequencing. And I want to highlight these because many rare conditions require genetic testing in order to lead to a diagnosis. There are some conditions out there that you can diagnose clinically, but for the most part, especially in the rare neurodevelopmental world, we need genetic testing in order to get to a diagnosis, which is why genetic testing is very, very important and will be a point that I highlight many times during this talk. And as I mentioned, not all genetic variants lead to a genetic condition, and we have a classification system that we use to help us determine the likelihood of a variant to lead to a genetic condition. You'll see kind of 5 categories here, and if you looked at a genetic test report before, the ones you've probably seen are the bottom 3, so VUS, likely pathogenic, and pathogenic. I want to highlight VUS because I recognize that, kind of as the name suggests, uncertain significance, these are variants that we're not entirely sure whether or not they're leading to a genetic condition. We just don't have enough evidence to support it in either way. And I realize that this could be challenging for some families if they get a VUS back on their report because they're kind of left in this kind of gray area. There's a lot of uncertainty there. But I do want to mention that it is possible for VUSs to be reclassified over time as we acquire more evidence and we learn more about the specific gene or protein that we're testing for. It's possible that these can be reclassified in either direction. So for example, if we have a VUS on our report, but over time we see that, oh, this variant is popping up in various healthy individuals that don't have this condition, then it's likely this variant isn't what's causing the condition, and we can downgrade it to something like likely benign or benign. Conversely, if we see more patients that show up that have similar features of the condition that have this variant, then we're generating more evidence to say that, oh, we think this is actually associated with this condition, and we can upgrade the variant. Switching gears a little bit to talk about rare disease more generally, a phrase that I really like is that rare diseases are individually rare but collectively common. I think that's a nice sentiment to kind of carry out. And we have statistics to back this up. We know that roughly 300 million people across the globe are affected with a rare disease. And specifically in the US, where we define a rare disease as one that impacts fewer than 1 in 200,000 people, 1 in 10 people in the United States are affected by a rare disease. So again, individually rare, but if we think of them together, collectively relatively common. And we can group rare diseases by different types of similarities. So for instance, what organ system is involved in the condition. So for rare neurodevelopmental conditions or NDDs, this is typically the brain. Which cellular pathways are involved, which genetic mechanism involved, meaning what's the root cause of the disease. Is it that the protein simply is not made and there's not enough, or that the protein is still made but its function is altered? It's important to understand the genetic mechanism and the root cause. And some more statistics. This is a graphic from Global Genes. You may have heard of this nonprofit organization before, but they help support rare disease foundations. And there's a few on here. There's some on there that I wanted to highlight. Let me go back. So 1 in 2 rare disease groups don't have a foundation research supports group. So this puts ZTTK ahead of the curve. Having a foundation or a support group for resource for families and kind of a beacon for other researchers in the field is a huge advantage. 8 in 10 rare diseases are genetic. So again, this is getting at the importance of genetic testing and needing to do genetic testing to get to a diagnosis, because we see the average time to get to a diagnosis is far too long at 4.8 years. So it's really important to advocate for genetic testing kind of from the patient and family perspective, but it's also important for genetics professionals and policymakers to get creative with meeting this huge demand for genetic testing, because we simply don't have enough of us in the field to be able to meet that demand. So it's going to take some creativity to try and catch up there. And 95% of rare diseases lack an FDA-approved treatment. So there is a huge unmet need where we're getting better at being able to associate genes with genetic conditions, but we're not moving as quickly for developing therapeutics, and we'll talk about why a little bit. And this is just another slide to show kind of on this graph that you're seeing from a resource online that categorizes rare diseases. That graph has gone way up, but our therapies are still very far behind. For example, we have almost 6,500 rare diseases that have been defined, at least in this one database, but only 500 or so have an available FDA-approved therapeutic. So it's a very large unmet need. So some key takeaways from part 1. We know that ZTTK is caused by pathogenic variants in the SON gene. I hope I've made clear that access to genetic testing is very critical, and there's a major gap between disease gene identification and therapeutic availability. So how do we get and address some of these key takeaways, specifically understanding the SON gene and its function? We do research. So to go into some basics, I want to start from this graphic that is from our center, the RSZ TNC, that really kind of goes over our thought process and approach for research for rare diseases. So first, we start with genetically defining a cohort, and we say genetically again, as I said, genetic testing is very important, and to define a cohort, you need to have individuals doing genetic testing to find out they have changes in the same gene. And then next, you want to really deeply characterize the phenotype, which just means the clinical features of the condition. And we can do this through different types of research studies, which I'll talk about. And then points 3 and 4 are referring to a lot of the work that's done before you get to a clinical trial. So we call this preclinical research. And this can involve things like developing cell lines, animal models. It's where you can identify things called biomarkers, which are kind of hallmark features of a condition that you can detect, for example, on a blood test, things you can potentially detect on imaging like MRI or EEGs if there's a unique signature that is associated with the condition. And then in point 5, we get to clinical treatment trials, and this is where treatments are tested for their efficacy and safety in the patient population. And a huge question that we get asked a lot is, why does research take so long? Why is it such a slow process? And I get it. It can be frustrating, especially from a family perspective. If you're not on the inside of the research world and you're just looking in, it can be frustrating about how long it takes. And if you think of the figure I just showed, it looks pretty simple. We know what the steps are. We know how to get to the end result. But a more realistic look at what the research process is like, specifically in rare diseases, it looks like this. This is the rare research roadmap. Again, another figure from Global Genes. And you can see it's a windy, bumpy, turny road that has a lot of speed bumps and checkpoints. Something big you'll see on the right side of the screen here is the big try again sign, which unfortunately is very common in research, where maybe you'll have a treatment you've identified that looks really promising in an animal model, but once it's introduced to humans, it may not be as effective. So you have to go back to the drawing board. Unfortunately, this happens a lot. But there are a few things that I'd like to highlight on here, and this looks like this might be an old version of the slides, but down in the bottom left-hand corner is something called the Researcher's Toolbox. And in there, we have things like animal markers, or animal models, biomarkers, cell lines, iPSC cell lines. These are all the tools that researchers can have in their toolbox in order to advance their research. And two things on there that I'll point out are patient registries and natural history studies. I'll go into much more details about those soon. But first, I want to point out the drug repurposing sign here. There's another talk coming up on that later today, so I don't want to steal Richard's thunder. But I just wanted to briefly introduce this as a strategy that's being used more commonly in the rare disease world. So what it involves is looking at different drugs or compounds that already have FDA approval, but for a different indication, and trying to repurpose those drugs to be used in your condition. Again, there's more on that later today. So back to natural history. So what is it? It's defined as the course a disorder takes in the absence of treatment over the affected individual's lifetime, and it's very important to characterize and understand the natural history of a disorder. Kind of what Ada was saying earlier, you need to understand in order to make progress. And why do we want to study it? You know, it gives us the full picture of a disorder. It can inform clinical trials. We need to know what the features are in order to know what to treat. But two key things here are, it also helps families know what to expect. You know, in the rare disease world, unfortunately, there's a lot of unknowns and a lot of uncertainty. And if we start to study the natural history of a disorder and group patients together and see what their features are, how they change, how they evolve over time, it can help families give them a general idea of what to expect for their child or loved one. They also can help us understand what the impacts are not only on the affected individual, but on the family themselves. We know that caring for somebody with complex medical needs is a challenging job, and it can take a toll on families. And oftentimes we see parents and caregivers putting the needs of their children before their own needs. And we want to understand what those broader impacts are, and doing a natural history study can help us elucidate some of those. Sometimes you'll hear natural history study and registry used interchangeably, but they are different, so I want to point that out. Again, a natural history study is a pre-planned research study. How we collect the data, what type of data we collect is all pre-planned with a common goal in mind, whereas a registry is more of an umbrella term that just refers to collecting information about a specified group of people. A registry you may have commonly heard of is called the contact registry. And going into more detail, again, natural history studies are intentional in that they're designed in order to help us uncover, a lot of the clinical features of a condition, and also to inform future research studies and clinical trials. They have a lot of different types of components, a lot of different ways that we collect data. One that I want to highlight here is biospecimens. I know we have some biospecimen collection going on, and I really want to encourage people, if they feel comfortable giving biospecimens, to do so. These are incredibly helpful tools to have in research because they can really help us uncover the underlying biological mechanisms at play for a disorder. It can help lead to things like cell lines. So these are really powerful tools to have in your toolbox. And data in natural history studies usually is some combination of data that's collected directly from parents, so parent-reported data, and then also physician-reported data. So it's nice to have both to be able to kind of make comparisons between the two. And registries are a useful tool because they compile a lot of information into one place. If your registry has basic information like demographics, contact information, maybe a few key clinical features of whoever's enrolled in the registry, this can help identify people more easily who may be eligible to participate in research and clinical trials. In terms of study design for natural history studies, there are kind of two main categories. First being cross-sectional. So this refers to kind of a one-time study visit setup where patients and their families come in for one study visit. You can collect historical data, so kind of what their medical journey has been up until that point. And these are a great starting point, especially for new disorders where natural history hasn't been fully characterized yet. But the most ideal study design is longitudinal, meaning we're following patients over time. And this allows us to see not only what the features are, but how they might evolve and change, and if there's anything new that could come up as individuals get older. And they help inform what are called endpoints, which I'll talk more about later. But essentially, endpoints are what ends up being tested and measured in clinical trials. And having longitudinal natural history data to inform that can really be powerful for progress. And switching gears a little bit from the science to some ethical considerations, which I'd argue are just as important, even more important as some of the science. I've heard that there's a really big conversation happening in the ZTTK community right now about what the word cure means. And I want to point out that this is a super important conversation to have because that word is going to mean something different to everybody. I think that some people may be completely offended by the word. Some people may be fully motivated by it. But I want to point out that there's a lot of common ground where this conversation is happening. We all love and care and advocate for our children or our loved ones who have ZTTK. And although there might be some differing opinions, I think it's important to highlight, all of these opinions are coming from a place of love and care. So I hope that's, something that's a little bit uniting when we may have a lot of differing opinions on what something like the word cure can mean. But there are other conversations that are important to have, what the risk versus benefits are, especially once you get to, potential treatments. Are the risks outweighing the benefits? And then ensuring equitable access to research opportunities and to treatments. Somebody already asked about, if you're an older individual or a child, will that limit your eligibility for participating in something like a clinical trial? So having these types of conversations is really key to go alongside with some of the scientific progress to make sure that your research is, ethically guided. So endpoints, as I mentioned, are crucial to clinical trial success. They're what we're testing and measuring. To define them a little bit more in detail, these are how patients feel, function, and survive. We refer to these as direct endpoints. An indirect endpoint would be something like a biomarker. And it's really important to ensure that your endpoints are meaningful to the patient population, which means we need to hear directly from the community what matters most to them. There was an interesting piece of data that came from the ZTTK survey that I know recently went out. There was kind of an expectation that given how prevalent seizures are in the community, that seizures would be kind of top of the list, number one priority. But that wasn't the case. It was speech and cognition. So kind of highlighting why it's really important to engage with the community, to hear what's important to them, so that way our research can be guided by what's meaningful. Who pays for research? Traditionally, this has been the government, private companies, nonprofit, foundations, philanthropy. I know government-funded research is a bit of a hot topic now, so I won't go into too much detail there, but this is really just to highlight who the key players are and who to try and form partnerships with if you're looking for funding. From a foundation perspective, foundations can fund research through through fundraising campaigns. I'm not a fundraising expert, so I won't go into too much detail. But one thing that I want to highlight that at least we found is really powerful for a rare disease community is to have somebody, a researcher or a physician who can really be your disorder's champion. Somebody who invests a lot of time, makes a career out of studying your disorder, which is why it's so great that we have Dr. An here today with all of her amazing work. Having a champion and really investing in people who care about your disorder, I think, is something that's very, very valuable. I want to show quickly the last few things. This is a framework that our center uses to evaluate a given gene and disorder pair. Some genes can cause multiple conditions, so we pair a gene with one specific disorder, and evaluate the suitability of that gene or disorder for gene-based therapeutic development. In 2022, Maya Chopra, who will be here later, and her colleagues at the RSZ TNC published what we call the Gene Target Framework, and Gene Target is an acronym which I'll show up on the screen in a second. But we think that this could be a potentially powerful tool to use to really evaluate your translational research pipeline. And we evaluate based on these 10 criteria. So things like genetic mechanism, understanding what the underpinnings are, natural history study is on here, tools to deliver to target tissues at the right time. This is seeing what biological techniques or strategies can be utilized to ensure that you're targeting the right tissues that may be impacted in your disorder. We have ethics on here. A lot of these preclinical studies, things that would come from animal models, things like reversibility, to see if, in an animal model, in a cell line, if the compound, say, you're investigating, is it changing any of the phenotypes or clinical features that you're interested in changing? And again, we think this is a helpful tool to be able to kind of zoom out, consider all of these different things, and see how your pipeline is evolving. Are there gaps that you can fill in? Are there different places that you could target funding? All these things. So some key takeaways from part 2. So understanding the natural history disorder is really foundational to making research progress. It's important to zoom out and consider the large research roadmap, to consider the circular figure I showed earlier, to look at your translational research pipeline and see where your gaps and strengths are. And we're seeing that foundations are playing an increasingly critical role in accelerating and shaping research. And it's so important to have foundations engaging with the research community, know what to research. We want to know what's important to the community. These are some of the wonderful people at the RSZ TNC that I get to work with. My email is here on the bottom if you'd like to get in touch with me. It's very, very long. I apologize about that, so I'll leave it up there for a second. But it was a great honor to be here today. I'm so excited for you all to have this conference. Again, congratulations to you all for being here. I think it's a great, great accomplishment. And happy to take any questions if there are any. Thank you all.